Okay, the analogy for we need a lab means that we need tools. And so tools for AI are different types of foundation models and other types of tools or data sets and knowledge bases. And second, in order for scientists to use tools, we need then large language models that can orchestrate and work with those tools. So the purpose of today's talk is for me to show you how we are making progress towards realizing this vision, but by A, developing tools that serve as a lab that scientists can then use, and second, developing scientists that are AI-based scientists that now can use those tools flexibly. So first, there are a number of models under the AI paradigm that in the group we are developing, and those span 4 biological scales. So we think about molecules and molecular models that can help with reasoning and understanding specifically what are the functions and the role that different molecules have in specific biological contexts. Second, we know that molecules don't operate independently of each other, but rather instead they form molecular interactions, and that those molecular interactions then are embedded into broader cellular environments, which are defined by cell states and cell types. And finally, the 4th scale is that of reasoning at the level of tissues or entire human patients and human populations to get a sense of how well effective and safe those candidate new treatments are. So I want to share with you a few vignettes of the models that we're developing across these 4 scales. So first is that of molecular scale. And the motivation for our research in this area is a broad observation that as a field, we now have quite a number of highly powerful models that can model and predict and characterize protein structure from primary amino acid sequence. And for important classes of protein families, they can achieve atom-level accuracy or match that of experimental efforts. And that has been tremendously important for the field at large, and it paves the way towards realizing this grand challenge of having models that can predict and characterize the structure for every protein. There is, however, another yet grand challenge that we think is very important and remains largely open, which is that of modeling understanding functions or phenotypes that those proteins are involved in and play a role in, which is crucially important for characterizing, identifying, and assessing targets, among others. So we embarked on this problem and challenge of developing foundation models that can help provide functional insights for a very large, broad regions of human proteome. Why would you want to do that? We would want to do that because still 20% of the human proteins lack functional annotations. Very practically, that means that if you go to SwissProt, UniProt, other canonical databases that you typically use, that we all use to explore functional annotations of proteins we are studying, they're just empty pages, or they're very poorly functionally annotated. 95% of all publications in life sciences focus on 5,000 most well-studied proteins. That means that simply by reading literature alone, we are just not able to cover these vast regions of the human proteome that remains inaccessible to models that would just read PDFs of our papers. So we developed an open source and model that we refer to ProCyon. And what ProCyon is, it's a multi-model foundation model that can predict, characterize, and generate phenotypic descriptions. ProCyon operates at 5 different levels or 5 different knowledge domains that are represented here by different rows and relate to modeling and understanding, predicting biological functions, processes, and cellular components. Second, in reasoning about the roles of proteins in disease mechanisms. Third, about the roles of proteins in drug mechanisms. And fourth and fifth, understanding of functional impacts of molecular interactions and functional impacts of substructural domains in proteins. When the model is trained, human scientists can interact with the model in plain conversational mode. And there are 3 types of interactions or modes of collaboration with human scientists that ProCyon supports. First, what is shown in panel A is an example where human scientists describes in their input prompt the biological process of interest. They describe the task that they want the model to solve, and then the model will look at that biological process and then be able to scan, retrieve, score, and rank proteins that likely would satisfy the task described in the input prompt. Second, the model can engage with the user in an interactive back and forth yes and no session to have a conversation or do question answering about a specific protein. Importantly, the user input prompt here can be natively multimodal, meaning that user can interleave textual description of biological function with structure and or sequence for the protein they would like the model to operate with. And third mode is a generative mode. Here the model is asked to generate new outputs. And the outputs that we are generating here are not structural outputs, but text, phenotypic descriptions. We call them sometimes protein caption cards that describe in a conditional manner what a protein that is in the input prompt actually does. So this is how you can interact with the model when the model is trained. So how do we train the model? To train this model, there are two things we need. We need the data set, and we need the model architecture that we will then train using the data set. So first, the data set. To train ProCyon model, we created and curated ProCyon training data set that is openly available. And it's a data set of 33 million data points, where each data point is a tuple. And you can think of a tuple as having two elements, where the first element is protein ID, and the second element is a paragraph of text, a passage of text describing a particular function for that specific protein. In addition to this multimodal protein text data set, we also have information about primary protein amino acid sequences, information about predicted and experimentally derived protein structures that are used to train protein language, and protein structure encoders. And finally, a large number of scientific natural language in order to equip the model with the ability to understand and converse with scientists in plain language. Given this data set, we use it to train ProCyon model, and architecturally speaking, what this slide shows is at the high level key modules or components of ProCyon, which we can read from left to right. And we can see here on the left are sample prompts, which can be either retrieval, predictive prompts, or generative prompts. And those prompts can combine different types of modalities. They are decomposed by the model into text, structure, sequence-based components. And these three different modalities are processed by modality-specific encoders that have additional projectors, and those projectors ensure that the latent embedding space that is unified across these three different modalities captures the biology and the structure of the training data set. Then the model can either decode and rank score proteins from the unified latent embedding space if it operates in the retrieval mode, or if it operates in generative mode, the model can use autoregressive sampling strategies to generate text conditioned on the input prompt that the user specified. So we train this model architecture on the data set. We use a composite objective function that ensures that the model can follow human instruction, that the model can retrieve correct proteins and rank them very highly, and also that the model can generate text that is coherent to read. And altogether, trained a model that has 11 billion model parameters that we made available to others as well as to analyze ourselves. As we usually do when we train large-scale foundation models, before using the model for anything new, what we typically do is to use the model and benchmark it across established benchmarks to see how well it performs. There are probably 30 to 40 pages of these type of plots that we have in supplementary information for our paper. So I'm just showing here one snapshot of those benchmarking experiments where we have tested ProCyon in a few short setups for different functional categories. These are different bar plots. And for each bar plot, we measure performance in this slide as Fmax score, higher value indicates better retrieval. And we compare ProCyon, shown here in purple, to a variety of existing models, including models that are unimodal, state-of-the-art models, like state-of-the-art sequence-based protein language models, or state-of-the-art structure-only based models that are atomistic models. And then also we compare them to emerging models that are multimodal and combine text with sequence and structures, such as BROT-ST or Biotranslator, and also include more standard bioinformatics pipelines that use BLAST and k-nearest neighbor on top of BLAST sequence similarity in order to predict protein functions and other functional categories. And we are seeing broadly across many of those categories ProCyon performing really well, which has encouraged us to also go and use the model in some interesting novel ways. So here are a few vignettes of that. We've been working with collaborators who are studying Parkinson's disease and became very interested in protein ACAN-AD1, recently implicated in the onset and progression of Parkinson's. But if you look at AKN-AD1, it's a stop. So in canonical functional databases, it's poorly functionally annotated. So we just used ProCyon and asked ProCyon whether it can generate this type of conditional phenotypic descriptions, and it did so. And here are some examples of what it generated for AKN-AD1. And so one of the phenotypes that it generated was about the involvement of AKN-AD1 information of actin cytoskeleton, which we then independently validated through subcellular localization assay for that protein in the Human Protein Atlas. Here's another example of how our collaborators from Imperial College London and King's College London used ProCyon in order to study and characterize targets of multiple sclerosis. And so collaborators are very interested in very precise, deep characterization and understanding of those targets that they could map to specific pathology in MS. So what they came up with, their neuropathologists wrote down these 5 distinct types of descriptions of MS pathology that is clinically relevant for MS. And so initial parts of those prompts are shown on the far left of the slide. And each of those prompts was sent to ProCyon, and ProCyon was asked to retrieve and score proteins that would be most characteristic and could explain collectively that specific type of pathology. And so what we have done the next, or our collaborators have actually be able to stain 280 samples of post-mortem brain and perform bulk RNA-seq analysis on those, and also classify them to each of those 5 distinct pathologies. And we looked at what is the overlap between those differentially expressed proteins in a pathology-specific manner that were identified through RNA-seq analysis, and those proteins that were retrieved highly by ProCyon and were assigned high percentile ranks. And what we can see here is that across the different pathology types, there is a substantial enrichment where those proteins retrieved by ProCyon were indeed characteristic in experiments. And so this is an example of kind of information leakage proofed type of analysis for the model, where it was used in a forward direction to test novel hypotheses it made. ProCyon can reason about one molecule at a time. And specifically, it can take as input one protein, potentially multimodal data about that protein, and it can reason and provide some functional insights about that protein. And now we know that those proteins don't interact independently of each other. So we've recently been developing a model called Atomica that is focusing specifically on this question of modeling not one molecule at a time, but modeling interactions between those molecules. And so what Atomica is, it's an all-atom-based model of molecular interactions, which is specifically focused on taking interaction complexes and representing interaction complex with a graph, and so which is shown in the center of the slide. In that graph, we have nodes that represent atoms, or nodes that represent chemical motifs, residues, higher-level structures. And edges between those nodes indicate bond lengths, the hydral angles, and other important properties. We then create and build this type of graphs for the entire data set of 2 million molecular interactions that span different molecular modalities and cover protein, small molecule, protein, RNA, protein, DNA, lipid, ion interactions. The beauty of this approach is that because it's atom-scale approach that operates at the level of atomistic graphs, we can train a single unified model across these different molecular interactions. And we train a model through a pre-training objective that is a one-step discrete diffusion objective. You can effectively think of them as an objective that masks specific block in the input graph and trains the model in order to predict what is the identity and the structure of the block that was just masked. So it's type of language modeling, but not when we have a sequence of words, but it really operates in this 3-dimensional space. What can we do with this model? Again, we did lots of benchmarking. I will skip all those analyses, plus some interesting compositional structures of the embedding space that we can show. But the PhD student here, Ada, who has developed this model, she was very interested in this idea of dark proteome and whether we can illuminate dark proteome and identify and predict functions related to molecular interactions for some of proteins that are poorly characterized here. So she used Atomica to nominate 5 potential heme binders. And so then our collaborators from the chemistry department over at MIT have synthesized those proteins and then ran functional assays on them to confirm that those predicted heme binders were indeed heme binders. What I find most exciting here, however, is that 4 out of 5 of those predicted heme binders could not be identified using standard approach. Standard approach being taking a molecule and scan it for the presence or absence of certain structural motifs that are indicative of heme binding. So this is an example that illustrates of how these models, when used thoughtfully and carefully, they can also provide insights that go beyond what traditional models can allow us to do. So now that I'm 2 minutes over time, so I'll just wrap up with 3 slides, perhaps, if I can squeeze them in. So I talked about models. So models, you can think of them as analogs for AI of what tools are in a wet lab, a scientific experimental lab. Well, but tools are not enough. There should be people who operate those tools and who generate interesting research plan and execute them. And so that practically means that there's another component that is missing, and specifically we need now orchestrators that will use these different types of foundation models across different types of data and different types of modalities. So we have built exactly that. So if you go to aiscientist.tools, you will see ToolUniverse. ToolUniverse is a universe of 600 plus scientific tools that cover a broad range of ML models, software packages, bioinformatics workflows, tools for automations, knowledge sources, RAG systems, visualization, et cetera, And what ToolUniverse allows you to do is to connect your favorite large language model, which can be closed or open source model. It can be Claude, it can be Gemini, QN, GPT Codex, or something else, with that ToolUniverse, so that what you then get is an interface like that if you connect it with Claude, where if you go down, you can see ToolUniverse, 600 plus tools, and you have your scientists that will now be able to use the tools that are in ToolUniverse. And this is made possible by kind of novel advances that relate to AI interaction protocols, where models can interact with each other and interact with other types of tools that human scientists use. So with that, I'll wrap up here. Acknowledge a fantastic group of PhD students and postdocs and research associates in my group, and happy to take any questions.